Well, hi, everybody. Great to see you all here. My name is Jennifer Doudna, and I want to welcome you to our panel discussion entitled CRISPR 2.0, The Next Generation. So today's conversation is going to explore the powerful genome editing technology called CRISPR and the potential to revolutionize medicine. And as a little brief background, CRISPR is a technology that enables scientists to precisely change DNA, change sequences in the genetic code that can alter the function of genes or even cure diseases, as we'll discuss. And what's happened over the last roughly 12 years since the first academic paper was published on this in 2012 is that many teams, both academic and companies, have been developing CRISPR as a way to intervene, potentially creating cures for genetic disorders. And that's something that we're going to discuss today in terms of where the field stands now and where it's headed in the future. I'm delighted to be joined by Sam Kulkarni, who's the CEO of CRISPR Therapeutics, and by Benjamin Oakes, who's the CEO of Scribe Therapeutics. As you may know, Sam and his team made global headlines late last year by bringing to market the first FDA-approved CRISPR-based therapy for sickle cell disease. And Ben and his team, yes, let's start. Very, very exciting. Ben and his team are working on the kind of next generation of CRISPR and figuring out how to do in vivo therapies that could address disease at scale. Both of these leaders and their teams are pushing the boundaries of what's possible with CRISPR technology, and they're opening up our ability to permanently cure and not just treat disorders. Incredibly, all this progress has happened in just over a decade. I think the era of CRISPR medicine is just beginning. We're also at this remarkable moment where we can see the potential and the opportunities, and yet there are some challenges that we'll get into. It's also really a testament to the power of fundamental research, as well as the dedication and the collaboration of thousands of committed researchers, doctors, companies, and brave patients that have been participating in clinical trials. So let's discuss where things stand and what challenges we need to overcome to make new breakthrough CRISPR-based therapies more accessible and patient-friendly. And we're going to speak for about 40-ish minutes, and then we'll turn it over to questions from the audience. So let's dive in. So Sam, I'd like to start with you. There are thousands of diseases. Why did CRISPR Therapeutics choose to pursue a cure for sickle cell disease?
Yeah, thank you for having me here. It's great to have this discussion with you, Jennifer, who started the whole field a decade ago. It's hard to believe that within a decade we've gone from the elucidation of a new technology to having a commercially approved drug. And it's hard to overstate the significance of having that first drug approved. I was having a discussion with some folks in Silicon Valley who were equating it to singularity. You know, we talk about AI and singularity when humanity becomes smarter than itself and what the ramifications might be. But the fact that we've learned to edit our own genome to make make ourselves better is that moment of singularity in a way. And also hard to overstate the impact on patients suffering from sickle cell disease. You know, it's for those who understand the disease or know the disease, it's the longest known genetic disease. You know, it was almost 70 years ago that we found out that a single base pair change in your genome, while it renders protection against malaria, could also cause a debilitating disease if you have that change in both your chromosomes. And there hasn't been a single medicine that's been developed for these patients suffering from sickle cell disease. And these are patients who live a life of chronic pain, end up in the hospital all the time. And we're proud to have a drug that addresses this patient population and patients who suffer from severe thalassemia. So why did we start with this disease and this drug? You know, when we had this powerful CRISPR tool, it was, it felt like a kid in a candy store in a way, which was, there's so many applications we could go after. But what we didn't want to do is go after something that's so difficult that may take a very long time to validate the technology. And with sickle cell, having known the base pair change that we need to make, the fact that we're doing outside the body ex vivo, where we can characterize the drug product and understand anything that's happening at the genomic level in terms of off targets, et cetera, that was really important for us in the early going. So We almost said, let's go slow to go fast in the CRISPR space. And that's where we landed upon sickle cell. And it wasn't just us. There were many players who wanted to develop something in sickle cell, but it didn't go as fast. It's actually quite challenging when you think about a new modality of medicine. It's not like you're taking a pill every day or taking an injection. You're taking cells from a patient, manufacturing them, editing them, making sure that you have the right edit, and then transplanting that back into the patient. But we're really glad that with our partners Vertex, we've been able to get it to this approval, and we look forward to treating thousands, if not hundreds, if not thousands of patients.
Can you tell us how the CRISPR therapy works as a therapy for sickle?
Yeah, it's a very fascinating concept called fetal hemoglobin switching, and there are people in the audience I see here that started the field, like Francis, who started the field 40, 50 years ago. This was the notion, we're all born with a form of hemoglobin called fetal hemoglobin. And this is beautiful evolutionary mechanism where 6 months after we're born, fetal hemoglobin disappears and is replaced by adult hemoglobin. Some say that maybe because we wanted to run faster and adult hemoglobin can latch onto oxygen, release oxygen faster. No one fully understands the evolutionary basis of this, but we have a redundant fetal hemoglobin that we all have and that gene's present. And so what we found is that you can use CRISPR to make a modification of the genome that elevates fetal hemoglobin again. The same gene that was turned off by nature, we re-engineer it to turn on, and that can overcome the deficiency or defectiveness of beta globin, which is the subunit of adult hemoglobin. And this built upon years of work and from various institutions, including Boston Children's. People have seen the work from Stu Orkin and many of his associated scientists, and many others along the way that built on 40 years of biology. But with the advent of CRISPR, we could immediately address use this approach or leverage this approach for these patients in a very facile manner.
Can you tell us how the patients are doing? Any challenges that are coming up?
The data are quite remarkable. Almost every patient that's had Casgevy or exogamglutine autotem cell, it's a mouthful, that's the name of the drug, we call it Exocel. Every patient had benefit, and in fact, 97% of the patients in our trials were completely symptom-free. So you could call it these patients call it a functional cure. They describe it as a life before that where they couldn't imagine even having a job. We had a couple of people, I guess not patients, suffering from sickle cell disease working at CRISPR, and I remember they would have to go to the ER at a renowned institution like MGH in Boston, and people said, oh, you're just another opioid addict, want some opioids. And these patients were not getting the care they needed. They were living this life of pain. And in our trial, all these patients are getting benefit. In fact, they now say that they see a new life. We have our CRISPR offices in Boston where we engraved the quote from one of the patients, Jimmy, who said, this feels like I'm looking out at life at the new window, looking outside the window to all the possibilities, just like my child who's born now can see for the first time, can see grass or trees or anything else. I'm seeing life in new light. So it's very fulfilling that these patients get this benefit.
Extraordinary. So inspiring. Wow. Ben, let's turn to you. I want to hear about Scribe, your team's approach and your focus, and what are you doing that's different in the CRISPR space?
Yeah. At Scribe, we really are pioneering an approach that we call CRISPR by design, which is what we view to be a next-generation set of techniques and technologies and approaches that's focused on transforming CRISPR, which, as you alluded to in your talk earlier, is a bacterial immune system. It's something that we've harnessed from nature, and really transforming that into a more perfect genome editing tool by holistically engineering every single aspect of this for greater potency and specificity. So you can really think about what we're doing at Scribe as taking what nature has given us and reimagining it to actually be the therapeutic we want, so that we can start to imagine using these genetic medicines at scale, right? Our goal is not just to enable the treatment of rare disease, as we just talked about, and is critical, really, for CRISPR technology. but try to think about what technologies will allow each and every one of you to reclaim what I like to call our genetic destiny, right? We're all born with a genome. We all have, I would say, maybe aspects of that genome that are not as great, right? I know I personally tend to have a really bad cardiovascular disease in my family. And that's not something that I can get away from now, no matter how good I am from a lifestyle perspective. So Scribe's goal is to really use molecular engineering, a lot of techniques that I built and developed in your lab and at the IGI, to transform how we think about CRISPR tools from bacterial immune systems into really more perfect genome editing scalpels, if you will.
So there's a lot of buzz right now about in vivo editing. Can you talk to us about that? Tell us what it is and why it's exciting.
Yeah, I mean, obviously, I want to first recognize, what Sam and the folks at CRISPR have done is nothing short of extraordinary, right? Everyone here, you just call it a singularity, call it what you want. The fact that you guys have been able to execute so well to bring forward, genome editing, the original Cas9 technologies into patients at such a clip is something that, I'm a little bit flustered under the collar here sitting next to you, right? But it's true. It's nothing short of extraordinary. However, we all have to be honest about the challenges of the technologies as they exist. Taking cells out of the human body and then getting the human body to actually accept them back is a really difficult procedure. And it's really challenging for patients. So when we think about the next steps for genetic medicine, it is about how we bring these CRISPR genome editing tools into the human body, target a particular cell type that's associated with the disease, modify that, and really not have any other detrimental occurrences. So what this can allow us to do is create therapies that, again, are more applicable to much wider patient populations, create therapies that can treat diseases in cell types that you can't take out of the body. We can take hematopoietic stem cells out. We can't take neurons out. You can't really take hepatocytes out. And therefore, the future of genetic medicine being in vivo, I think, is a foregone conclusion already. And it's really about how we do that safely and effectively enough to be broadly applicable.
So Scribe is focused on cardiovascular disease, or that's at least one area that you've announced. What's the potential for CRISPR in that space?
Yeah, I think it is our stated focus at Scribe is to really, again, transform how any one of us can think about our cardiovascular risk. The easiest thing to discuss here, because most of you probably know of it, many of you are probably on pills for it, is bad cholesterol, right? LDL-C. There's over 2 billion people in the world who are currently not controlling their LDL-C levels, even though there are drugs that if you take them every day, and I mean every day, it will lower your cholesterol significantly. What this means in the real world is that about 50% of patients or less are able to stay on those drugs. And this results in a really dramatic increase in the rate of cardiovascular disease. You kind of roll this all the way down, and cardiovascular disease is the leading cause of death globally. It actually outstrips cancer almost 2 to 1, yet we hear a lot more about cancer. And I think that at Scribe, we chose cardiovascular disease because there's really well understood biology as well as pharmacological interventions that have proven to really modify this disease, but yet we don't have medicines that can enable these modifications to be lifelong, which is what you need. So we're really focused first on modifying LDL-C in any patient such that you can just get that taken care of, right? You walk into your doctor and you walk out a healthier person. I think that vision is one that requires genome editing tools that are much more potent and much safer than what comes out of the ground or what comes out of, naturally we find. And in the end, if we're able to accomplish that, we can imagine lowering the rate of heart attacks by 10%, if not more, in any healthcare system that exists today. I mean, there were studies that have shown if you could create a statin, which is an LDL-C lowering drug, that patients would just stay on. You would save the United States healthcare system $25 billion And that's in 2015 money. So today it's like probably $1 trillion, right? But that's a meaningful amount of money for any healthcare system. And when you start to think about applying this from a public health perspective, if we could lower LDL-C really broadly, we can really start to bend the curve in terms of what the number of or the rate of cardiovascular events is globally, which I think is really exciting.
Really exciting. Amazing vision. Let's turn to discussing a bit about access to this technology. And Sam, I want to turn back to you, and maybe you can speak to the, tell us why CRISPR therapies are so expensive, or are they expensive, however we want to define that. And also, how can we think about bringing down the costs and increasing access and making it more available to patients?
Yeah, if I may, I just want to piggyback on what Ben said about diseases as well. I would say that now not a week goes by where we don't find another application of CRISPR that could transform the disease. Every week, whether it's a common disease or a rare disease, a new idea comes up. An example is this recent publication on ApoE4, which is a genetic marker for Alzheimer's. I wouldn't be surprised if 1 or 2 people in this audience have 2 copies of ApoE4, which means you have a 10-fold higher risk of developing Alzheimer's, and not just developing Alzheimer's, you could develop it early. So what can you do to fix that? It's very hard to send a small molecule drug to do that. It's really genetic manipulation. So that's one example of one extreme where you're thinking about neurodegenerative diseases that you can completely prevent. Another example is, women's health. There's a disease called acute hepatic porphyria, and it's often undiagnosed. Generally women in their 30s present in the hospital in the ER with acute pain, and people think it's something else, and it's never diagnosed. It turns out there's a mutation in the heme biosynthesis pathway, which can be fixed with one edit. It's a very simple edit in the liver, we're trying to address that. That could completely take the pain away. So, every week we're coming up with newer and newer indications where we could have a profound impact. And I just think we're at the start of this. This is like the AI field where, all the work that NVIDIA did was happening 20 years ago to enable it, right? And then they all of a sudden hit this trajectory where now it's applicable across every sector. And that's what's going to happen in disease. I think it's hard to predict right now where the biggest impact is going to be. And we're looking at the biggest killers, obviously cardiovascular disease. Cancer's, again, a place we have to put effort in because it's 70 years since we declared the modern war on cancer. And we've made some progress, but we haven't really made a dent in terms of curing cancer yet, right? Or diabetes is another area we're going after. But I think we're going to see, I predict that if we're having the same discussion 3 years from now, there'll be many more indications which we could go after. Access is a different question, and I'm acutely sensitive to the access question. I grew up in India, a small town, and there's so many patients with rare diseases or common diseases in parts of Asia and Africa that are not going to be able to afford CRISPR medicines if they cost $1 million per patient. Now, it's pharmacoeconomically beneficial in the Western countries. And so, again, the question you said, well, are they expensive or not expensive, depends on which part of the world we live in. One of the reasons pharma is worried about CRISPR is because they lose the annuity. It's a one-time treatment that actually makes them less money than something that's an RNAi that we could give for 40 years and charge patients every 6 months. But on the other hand, there are parts of the world where this is not affordable at all. And so I think technology will solve that. On one hand, I think just the fact that more companies are doing it, there's going to be an improvement in the cost, just like we have with EV cars and other technologies over time. TVs now are, if you look at the, corrected real inflation values of different categories, education has gone up like this and TVs have come down like this, right? You can buy like a 100-inch TV for nothing now, which was very, very expensive 20 years ago. So I think technology will improve and cell therapy is going to get less expensive, but there's also profound step changes like in vivo editing that could really get it to places in Africa. We have a grant with the Gates Foundation to develop in vivo therapies for sickle cell. That could make it 10x less expensive. And even that's not enough for certain parts of Africa. So something we as a biotech have the freedom to work on to improve access. But it's important that the collective society works on it because, if you have a regulatory system where you have to have 6 potency assays and all the CMC characterization and everything else, it's going to cost $1 billion to develop a drug, and then it's hard to make it less expensive. And so there's something that requires the whole village to be working towards more accessible therapies.
Yeah.
Ben, how does Scribe think about making these types of therapies more globally available?
No, I mean, I would second that 100%. It takes a village. There's going to need to be, I think, real development on both the technological side as well as the regulation side to really lower the price of these therapies. How we're focused on doing it today is, of course, my background as a technologist, on the technology side. To me, every problem looks like a technology problem. Really, I think the best example of this is also in the sickle cell area. So we have a collaboration with a partner called Sanofi, where we are working on combining our genome editing technologies with improved potency and specificity with their ability to deliver essentially these genome editing components in vivo and target hematopoietic stem cells. Now, if we think about places like sub-Saharan Africa, or areas where there aren't major medical centers where you can essentially do bone marrow transplant, which is what's required for the therapies that exist today. Even beyond pricing, if we just think about access, you need to be able to deliver these things in vivo. And that's where we're really focused on enabling technologies that are maybe not as dose limited as the first generation, which again, this is a nuanced point, but the amount of CRISPR molecules that you need to get into the human body to have an effect is really important when you start to go in vivo. We can deliver a lot when we're out of the body. When we actually have to put it into a delivery vehicle and bring it in, we need those same molecules to be more potent per unit dose. And that's where we think we can start to bring technologies to the fore that might modify outcomes.
So one of the questions that came up in the earlier session about CRISPR was the issue of safety. We're talking about a therapy that changes DNA sequence. That's a pretty permanent thing.
Yeah.
How does your team think about ensuring the safety of the current therapy that you have approved, but also things that you might develop in the future?
Yeah, let me first contextualize safety because I sit on the boards of a number of companies. In the past 20 years, been involved in development of a lot of small molecule drugs and biologics. The safety issue, when you take small molecule drugs, you don't know all the receptors are hitting. You actually people ignore the fact that it's somewhat promiscuous in a way. So that's why you get all this.
Tylenol that everyone takes today is doing more damage to the liver than we're even allowed to think about.
Especially if you went out drinking last night.
Yeah.
There's toxicity with all medicines we take in a way. There's few. GLP-1 is a class of medicines that 15% of patients get nausea, but it is a beautiful drug in a way because the tox profile's pretty amazing for a drug that can have such great benefit to all of us. But we'll see. It's early days. But with DNA-based medicines, ultimately, I think it's going to be seen as much safer because you're making a very precise edit to your genome. Yeah. And when we developed Casgevy, we tiled 1,000 different guides, did all the work to make sure there's no off-target effect anywhere. And yet, we had to go through, various rounds of discussions with US and European regulators. In fact, we had to go through an advisory committee discussion, in spite of showing that it's very safe. But I think we wanted to be very responsible as the first drug that's going into an approval process to make sure we develop it that way. And the same philosophy applies for every other drug we're developing. Now, in the cancer setting, it's a little different. I think, you have a different risk-benefit analysis because you're using cells that eventually get eliminated anyways after they kill the cancer cells. So it's not as big a risk. But over time, I think that's the beauty of CRISPR is it's very directed. This is a good ChatGPT exercise, which is if you were to tell ChatGPT, here's what CRISPR can do, can you develop an illustration to describe CRISPR? If you did that, 7 years ago, and we didn't have ChatGPT, so we asked an actual illustrator to do it. They came up with the image of molecular scissors, right? That's what CRISPR was. But if you do it now, the image that comes up is a drone that can deliver the exact effector protein to any place on your genome in a very specific way. Most of your passwords, I'm pretty sure in this audience, people use passwords that are like 7, 8 characters. This is a 20-letter password that's super secure, right? It's only going to one place on your genome, and you're using a drone to deliver right to that place and then make a precise modification of the genome. So I do think it's going to get even, people are going to buy into the safety more and more as people see more data. But it does take companies at scale to do it. I'm aware of certain people who are trying to do investigator-led trials, et cetera. It's very hard to do what we've done as a company, to tile and empirically identify that there's no off-target in thousands of guides before choosing one. But it's something that we all need to watch very carefully, and something that's of highest priority for us at CRISPR.
You mentioned regulation and its role in ensuring safety. And Ben, I'm wondering, what do you think about the current regulatory approach? Does it support bringing to market these types of therapies, or do you think that changes are needed?
I think there's always room for improvement here, and I think Sam could speak to this as well, having brought these therapies all the way through to the end. We are all watching very closely how regulators react. And on this off-target question, I think it's one that's of utmost importance, right? I want to second what Sam just said, right? When we think about being able to quantify the potential off-targets, we have never been able to be, at least in my mind, as careful as we can be with CRISPR, because we can read out what is potentially happening off-target on the genome. Meanwhile, all of these other drugs, you kind of look for general safety. We gave it to patients, patients seem healthy. And then therefore that's okay. With CRISPR, we actually have to go in and quantify across hundreds to thousands of different sites. We see editing or we don't see editing, and at what dose. So we are, I think, held to a higher standard by regulators, which is an interesting conundrum, right? We have a more specific technology, a technology that is potentially more transformative than any technology before, yet just by nature of it being so specific, you can hold us to a much higher standard. And I think that will be something that we have to kind of wrestle with as we start to think about treating more and more diseases. What level of comfort can we develop with these sorts of technologies? And can we start to move maybe more in the direction that the regulators have started to talk about, which is with more of a platform designation, with an understanding of if a tool is working in a certain way time and time again, the regulatory burden gets lower and lower. But one thing that I think you'll always see is the regulatory burden of manufacturing, and the manufacturing of these technologies tends to be a little bit more complicated given the numerous number of components. So I think that there's certainly a ton of room for improvement, but it is, I think something that we can also really start to think about overcoming with additional improved technologies. And I'll actually stop there, but I'm happy to talk more about the improved technologies if you want me to.
So given the landscape, the challenges, the technical and scientific advances that are still in progress to make all of these things a reality, I'd love to hear what you each think are the role of academia and nonprofits in helping to bring CRISPR medicines to market and to establish a new standard of care. And maybe we could start with you, Ben.
I'll go first. I think I got the easy lead in here. So Scribe was founded out of an IGI, which is an Innovative Genomics Institute, which is the institute that Jennifer started at Berkeley, Entrepreneurial Fellowship. Scribe would not exist without that fellowship. That allowed me to start my own lab, bring a team of like 5 highly dedicated undergrads, basically, together to build technologies that have never before existed. What we've created at Scribe is actually built an entirely distinct CRISPR platform from anything that has come before. Many of you may know the original molecule discovered was called Cas9. The molecule that was later discovered is called Cas-X. And we've taken Cas-X and we've engineered that into something called the X editor. And that allows us to do much safer, much more specific genome editing. We've also taken that X editor and turned it into an epigenetic editor, and I'm not going to go there just yet, but there's some really exciting things we can do with that. All of that, none of that, I should say, would be possible without the IGI Entrepreneurial Fellowship. And so I think there's a really important role to be played at the very start of really transitioning, what is academic work towards work that's much more pragmatic, much more applicable. And I know, at least in my journey, that's been critically important.
Yeah, I do think academia is going to have a bigger role to play with CRISPR-based medicines than small molecules, for instance, because, if you had to develop a small molecule drug, you have to take a library of 1 million molecules, screen them. An academic is just not going to have the wherewithal to do that. Whereas with CRISPR, you can say, here's the gene I have studied for last 20 years. Here's the change I need to make. In fact, there's some people here in the audience that are doing that, right? Let's see if this works. And so you're going to see proof of concept on new ideas and new theses for how to apply CRISPR, whether it's cell therapy or gene therapy. The other place is rare diseases. I think for a lot of companies, it's going to be hard to make the equation work, especially if the regulatory environment is the same, to justify going after some of the ultra rare diseases when there's so much opportunity with common diseases. And I think that's where academics can play in as well. And the third place is long bets. What is the role that CRISPR can play in anti-aging? I don't know. This is an interesting exercise, by the way. I went to this think tank where they were talking about anti-aging and asked the question, how many people want to live to 150? All right, let's do that now. How many of you want to live to 150?
Why not? Why wouldn't you?
I definitely. It's very interesting. It always comes out to about a third of the audience. Okay, it's a third of the audience, plus or minus. It doesn't matter what setting you choose. And I don't know why that is. It'd be very interesting to live to 150. Like if I go back 150 years ago, we hadn't even coined the term gene. The term gene was coined about 120 years ago. So you get to see the whole evolution of how humanity lives. But there were two schools of thought, which is in anti-aging, you could either say, let's just do a lot of preventive medicine and prevent things like cardiovascular disease or neurodegenerative disease, and that's the way to live longer. Or say, just keep your brain intact and then we can replace every organ. Let's create mini pigs that can make artificial kidneys, create lungs. People are doing that, by the way. There's an amazing mini pig colony that one of our friends has developed over the last 8 years that can produce kidneys the size that humans need. And so, but those are things that are going to, again, I think, may have to be special institutions that are funded without the scrutiny of public markets, or I know someone asked the question about stock prices this morning, which is, it's very hard for me to justify developing something that's a 15-year bet sometimes, as a public company. But there's special institutions or unique industry-academia partnerships that can go after something so transformative. And I think you're doing some of that. So
we're trying, yeah. No, we certainly appreciate the need to have both. I think we really need to have focused teams that are really dedicated to particular applications and bringing those to market. That's not something academics do or have the wherewithal to do. But I think the innovation piece is something that AI specialize in. So yeah,
absolutely.
I'd like to turn to talking a bit about the future. Now, and AI and machine learning are hot topics in science and for investors these days. What role do you think AI is going to play in the future of CRISPR medicine?
Yeah,
either of you.
I can kind of start here as the molecular engineer, where AI has already shown itself to be very interesting, is in things like protein design. But it's interesting though. There's a lot of, dare I say, hype around that. And while it is effective, while it can do, I could generate today thousands of protein designs using AI. Guess what's next? They still need to be tested. And then when you test them, you still need to improve them. And then when you improve them, you still need to deliver them. It's so far upstream that I think of AI as being a really good hypothesis generation tool, and we use it at Scribe to start to think about ways we can improve our protein. But it's just one. It's not really a golden ticket or the magic bullet here. It is a really beautiful natural extension of what we have already been doing, a technology platform that allows us to kind of go beyond what we can do to proteins more rationally, but it's just that, at least at this point. And I'm sure Sam can talk even more about how many steps downstream are required to actually bring drugs through the clinic, and the roles that AI can play there. But it's like it's a tool in the toolbox. It's not, I don't know if AI today is going to dramatically improve or increase the number of drugs that actually get through the end stage approval 10 years from now. I think maybe some of them will be AI derived, but is it going to increase the overall number?
What do you think, Sam?
Yeah, so, 4 years ago, I was a huge skeptic of AI in medicine, and then I talked to some people who were deep in the AI space, and they said, let me explain AI to you. Let me use the analogy of American football. And I said, great, I didn't grow up in the US, perfect, let's do that. Said, okay, so let's say you're kicking a field goal. You can either do one of two things. One is train the kicker so that you get better and better accuracy in the kick, and the goalposts remain the same. Or you can just widen the goalpost because you have so much more confidence. You have so much greater confidence it's going to be a good field goal. And that's what's happening, I think, in genetic medicines. You can either find targets that are biologically validated through AI that give you that much more confidence that you can go after them. An example is a disease called NASH. Most people don't know about this disease, but it causes fatty liver. I know people who are like super skinny and runners, but their liver cells are very fatty, and eventually they all develop cirrhosis of the liver or fibrosis. And this disease, it's very hard to know what targets to go after, but there's this AI effort to identify any genes that may help with that disease, and that's actually working. Now, we'll see. Eventually, you have to do the trial to know that it works, but it's unearthed new targets to go after. So that's the example of widening the field goal so you have better confidence in going after that experiment and spending $500 million doing it. The part about training the field goal kicker to kick better kicks, we do some of that with mRNA secondary structures or trying to predict guides. So far, hasn't yielded amazing returns yet, but it is getting better because the key is you need to have more data. Unless you have more data feeding into the training models, you're not going to get better results, but that's happening, and I think eventually you're going to hit that hockey stick where you have a threshold of data, then eventually you may see a lot of improvements. But I do see quite a bit of new innovation in small molecules with AI and also with some of the antibodies.
Yeah. No, I think it's going to be very interesting to see how it's going to change our world for sure, right? It's just not entirely clear yet how.
What do you think,
Jennifer?
Jennifer?
Well, I agree with your last statement about data. I think this has been the thing that I've been really thinking a lot about is, yes, we have a lot of data in biology, but most of it right now isn't at a place or a quality level or a consistency level that we can use it for model training. And so I think that's an area where there's an opportunity. Yeah. Well, I'd like to maybe let's think about where this is all headed in the future, and I'd love to hear from each of you what you're most excited about as we look ahead to the next decade with CRISPR, but beyond that, what are the breakthroughs that you see coming along that are going to really transform the things that you're doing?
Yeah. Decade is an interesting question because a decade ago, or just a little bit over, CRISPR wasn't a thing, right? I started out working on genome editing tools back before CRISPR, right? So without being overly, I guess what I want to say here is it's incredibly difficult to predict just how rapidly we're going to be able to move things. My sincerest hope, though, is that we will have at Scribe, as well as others, developed a number of technologies that allow us to really bend the curve on cardiovascular risk. And we've developed them in a way that makes them safe and effective enough that pretty much anyone in this audience could go to your doctor and ask for it. I think that would be huge. I think we could, within a decade, really start to change public health from a cardiovascular risk perspective. And that's pretty incredible. I think I like to think about what my life would have been like if we had technologies like that. So my father has had 3 heart attacks, right? And he had his first at the age of 40. If we had had those technologies even 10 years ago, could we have prevented his second or his third, is a question I'm always asking myself. And I think 10 years from now, that's not a question that we're going to need to ask ourselves anymore. I think we're going to be able to take action on that. And that's what I really hope.
Yeah, I always think about things in the 10-year, 20-year, and 30-year horizon. I think in the next 10 years, we're going to see significant improvements in cancer medicines with editing or engineering of cells. We're going to see a lot of breakthrough data in rare diseases across, single base pair edits or multiple edits you need to make. The 20-year vision, I subscribe to what Ben's saying, which is there's going to be a lot more medicines that are preventive. You find risk factors, whether it's cardiovascular disease, whether it's diabetes, NASH, whatever else, Alzheimer's, and you're going to see a lot of prevention. And I think the 30-year horizon for me, and we're making those bets now, is replacing organs. I think you can replace blood, you can replace kidneys, you can replace heart even. And I think that's going to be quite transformative how we think about life.
Amazing. Well, I'd like to open the floor up now for questions. And we have a couple of microphone runners. And I will put on my glasses and see if I can actually see people and call on people. I saw your hand up first, right there, with the glasses. And can someone bring him a microphone? Yep, it's...
Thanks.
Francis, please. Can you introduce yourself?
Francis Collins, former director of NIH, and full disclosure, working intensively on a gene editing therapeutic we hope to be a cure for a very rare disease called progeria, which causes kids to age at about 7 times the normal rate. And it's caused by a single misspelling of one letter that should have been a C, but it's a T, in a gene called lamin A. Looking across the field, and you all have done a wonderful job of touching on many aspects of that. Thank you, Jennifer and Sam and Ben. For rare diseases, it seems to me we're making a lot of progress in the actual elegant ways to do the enzymatics. And we're no longer limited to the original double-stranded DNA break, which means you can knock out a gene. Sometimes you don't want to knock it out, you want to make it fixed. And so gene editing with things like base editing and then prime editing has come a long way. It feels to me, though, like the major difficulty with those 7,000 rare diseases where we know the DNA misspelling, but we don't yet have a treatment, is the delivery problem. Many of these affect the brain, so the idea of ex vivo is not going to be possible. It's going to have to be in vivo. And we're still depending on most of our clinical trials on a particular vector called AAV. And our regulatory system kind of insists on that because there's some experience there. And so if you're going to go to FDA with a trial and it's not AAV, you just bought yourself about 2 or 3 more years and tens of millions of dollars. So everybody keeps going with AAV. We are also. But we know AAV is not that safe. There are kids with Duchenne muscular dystrophy who have died from AAV therapeutics that went wrong. So are we doing enough to invest in the delivery systems? We haven't talked as much about them in this session. Getting away from just this single family of vectors. I mean, there's a zillion AAVs, but where are we with nanoparticles and lipid nanoparticles and other ways of doing the delivery that would be zip-coded to the tissue where you want the editor to go and would be safe and effective and could be approvable in less than 10 years by a regulatory system?
Well, I mean, I could start here. So I think there's something to be really, there's something to be thought about with delivery that's really important, which is delivery is not just a targeting problem. It's not just can I find the right receptor or the right cell type. It's like also a physical problem. And the thing that's great about AAV is physically it's small enough to actually get through a lot of the smaller junctions within our body. And that actually allows it to get to other places where a nanoparticle that's 70, 80, 100 nanometers couldn't actually get to. And like that's, I think a real challenge for delivery is finding the right combination of physical size, targetability, obviously safety, as you're alluding to. That is certainly not going to be a one-size-fits-all approach. So the question is, are we working enough on it? Yes. I think there are people who are working on it like every day, thinking about all sorts of new ways. Are we as a field going to find, again, the magic bullet? No. I think it's going to be, especially for a rare disease that targets many different cell types in the body, you might need multiple different types of delivery vehicles. And again, from a regulatory perspective, how is that going to be possible, right? Each one is its own manufacturing run. Each one may look very different, but I can get to the liver with an LNP, I know I can get to the heart with an AAV, and I know I can get to the brain with maybe something else. So I think that there are really huge advances that are happening in things like lipid nanoparticles. Again, I already alluded to one of our partners, Sanofi, who has the ability to now target them to specific cell types within the body. But that targeting is again, limited by where that lipid nanoparticle can get to in the first place. So there's a number of additional really exciting novel technologies that are coming online in terms of virus-like particles or things that harness other parts of different viruses. But to your point, I think they have their own challenges. Manufacturability of them is actually another huge reason why AAV is something we're stuck with, because we know how to manufacture AAV. It ain't pretty, but we know how to do it. And I will say there's also quite a number of really exciting advances in AAV. That's all to say that there's a lot of work going on, at least as far as I'm aware of, but there is no magical 5 years we're going to have it solved.
I'd love to have time for another couple of questions. Yeah, a hand right there.
Thanks so much. I'd like to go back to the financing question. Sorry, I'm a VC. Are there any things that you can think of that pharma can do today or in the next few years to whether it's partnering with employers, CMS, other institutions to make these more affordable? Or are there any interesting financing schemes you've read about or thought of to make cell and gene therapies more affordable?
Yeah, I'm happy to take that one. I'll take 5 seconds to just acknowledge Francis's support in getting Casgevy approved behind the scenes. It took a lot of education of regulators and NHLBI. A number of people there actually helped a lot, so thank you. And I do agree that there's a lot more investment going into delivery technology, so it does take a little bit of time. We entertained all options on the payment models. What's interesting is I think a lot of the U.S. commercial payers were actually said, we understand the pharmacoeconomic case. We'll just pay for it up front, even if people switch insurance, et cetera. It actually was less of a problem with commercial payers because the employers want more certainty, but it's more work for them to track what happens over 5 years, 10 years, et cetera, with these models. But all the government agencies are working on some experiments to say, is there a way to track patients and do amortized payments over time? And the Europeans certainly are looking at it very much more deeply because they have budget issues. So I think industry is very open to these models. It actually is much harder to implement than people think.
Yeah.
Mm-hmm. Yeah. Let's see. Question right here.
Hi, I'm Chloe Bird. I'm a professor of medicine at Tufts and senior sociologist at RAND. And I had a question for Ben on to what extent are the in vivo approaches helping us deal with persistence?
Persistence in what form?
In success in not only having made an edit, but that it lasts.
So I think, one thing about going in vivo you don't have to deal with is the re-engraftment issue, right? The re-engraftment challenge that you guys have solved, I think, in large part, at least as far as we know today. When we go in vivo, I think there's a lot of really interesting things that we can do when we talk about really enabling durability. One, a genome edit, if you can get to a cell that's really the cell that's going to be dividing, will be maintained. And we've seen that now ourselves in large animals out well past a year, complete flat line of effect in terms of no reduction of persistence of our effects. I think if I could actually take your question and turn it a little bit, we have newer technologies where we're actually not making a genome edit or a double-strand break or a permanent modification. We're actually using epigenetic control to silence a target gene. And we're doing it so in a way that should be heritable and lasts over time. But it really opens up this much more important question of just how long will this epigenetic silencing last. Within any human, your epigenetic code is one that is potentially degraded over time as we all age. And I think the question of persistence is much more relevant in that setting, where we're actually trying to silence genes, not permanently edit them. I think, excitingly, we've already seen some very promising work where we can not enable a genome edit, but still enable a very durable genetic silencing via epigenetics that also, on the flip side, is actually reversible. We can go in and reverse that epigenetics. So you can start to imagine now, if you will, follow me on this journey to a world where any medicine could be an epigenetic modification or I should say maybe any pharmacological intervention that currently exists, which are mostly knockdown interventions. Could be an epigenetic silencing that's a one-and-done therapy that you also could reactivate or turn back on. And I think this is where the future of genetic medicine really goes. It's a world in which we don't have to permanently modify our genome to have a durable outcome, to have a one and done. But we can, if we want to. We want to get, lower our LDL-C for the rest of time, we can do that. Or we can just kind of transiently modify and even tune the amount of gene product we want within our bodies. Incredible, I think, what we're going to learn from the standpoint of genetics and large-scale sequencing as well, in terms of really starting to recapitulate these phenotypes that are more tied to better health, and then use these technologies to maybe get persistent or actually, in some instances, non-persistent outcomes that might be what you want.
Fascinating possibilities for the future. I think we have to leave it there. I want to thank you both. It's been a fascinating conversation.
Thank you.
Yeah.
Thank you. Thanks, man.